Hello kids, today's video topic is endomembrane system. Endomembrane system consists of membrane-bound organelles. These organelles structure and function is different, but they work in close coordination with one another. These organelles are endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. So endoplasmic reticulum is membranous structure. Okay, it is consist of cisternae, tubules, and vesicle, and endoplasmic reticulum attached to nucleus and form nucleus covering. Okay, so this portion you can see this fold is this structure is endoplasmic reticulum, and it has ribosome attached to it. Okay, these ribosome produce protein, and because of ribosome, it look rough, so it is called rough endoplasmic reticulum. Now here in this portion you can see there is no ribosome attached to it, so it is called smooth endoplasmic reticulum, and it form lipid. Okay, so if you see a cell diagram, this is cell, this is nucleus, this is endoplasmic reticulum. This endoplasmic reticulum divide the extracellular space into two compartments. This space inside it is called lumen or lumen cavity, and this outside is called extraluminar space, where cytoplasm is found. Okay, so ribosome attached to the endoplasmic reticulum produce protein, okay, and that protein enter inside this lumen, okay, modified and secrete out in the form of vesicles. So both rough endoplasmic reticulum and smooth endoplasmic reticulum perform different function. First, rough, it synthesize protein and secrete it. It bear enzyme for glycosylation. Glycosylation means initial modification of protein, okay, is done inside the endoplasmic reticulum. Enzyme precursors formed with precursors are inactive substance which later form the lysosome. Now smooth endoplasmic reticulum synthesize lipid, where lipid is used in our body, like plasma membrane. Plasma membrane is made up of lipid. In animal cell, lipid like steroid hormone like estrogen, progesterone are produced by SER. So in it, these hormones produced in adrenal gland cortex. So adrenal gland cortex SER is more developed. It help in detoxification of drug. Like if a person take alcohol, okay, it detoxified inside the liver. So liver SER is more developed. Next is Golgi apparatus. Golgi apparatus is flat sac-like stained structure. Without staining, you can't see it. So Camillo Golgi first in 1898 saw it under microscope. It is made up of 4 parts: cisternae, vesicles, tubules, and vacuoles. The function of Golgi body is modification and packaging of materials. What materials? Protein. Okay, so here is the picture of the Golgi body. So this face, okay, suppose that this phase, okay, this phase which is receiving vesicles from the RER is called cis phase, and it is convex shape. Okay, and after entering inside the Golgi body, these vesicles, these proteins modified. So majority of modification take place inside the Golgi body, and then packed packaging in done, and then it secrete out in the form of vesicle. So this phase which is secreting this vesicles are called transphase. It is opposite to the endoplasmic reticulum, okay, and it is concave shape. So here you can see in this diagram, this is cell, okay, this is nucleus, this is endoplasmic reticulum, protein produced and modified little bit inside the endoplasmic reticulum, and the vesicles are secrete out. Then here is the Golgi body. Golgi body always located near to the ER. Also, this stacked parallelly, okay, Also their number is vary in different cell. Okay, so modification take place in both ER also and Golgi apparatus also, but majority of modification take place inside the Golgi body. So here, after the modification, the Golgi body secrete out the vesicles. A large amount of vesicles go outside the cell. Okay, some of them used inside the cell and some of them turn into lysosome and vacuoles. Okay, so let's talk about lysosome. Lysosome is product of Golgi body. Okay, Christian D. Dave in 1955 saw this lysosome. It is single membrane bound, a small vesicular structure, and it contain hydrolytic enzymes. Hydrolytic enzymes are digestive enzyme like lipase, protease, which digest protein, and carbohydrate, which digest carbohydrate. Okay, it is also called suicidal bag. Why? Because this lysosome secret enzyme and sometimes digest its own cell. That's why it is called suicidal bag. So function of lysosome, first it destroy bacteria like macrophage WBC. Okay, here is a bacteria. This WBC engulf this bacteria and form phagosome. Okay, so here is the lysosome which engulf it. Okay, and then destroy these bacteria. It removes dead cell and old organelle. Suppose that a cell is old and worn out. So here so many lysosome inside it will burst out, secrete enzyme and digest it own cell. Also old organelle, if and inside the cell any old organelles are present like mitochondria, suppose that this mitochondria become old, okay, not functioning properly. So lysosome will check this mitochondria and digest it. Okay, digest food like in protista, amoeba. Okay, here is the food. Pseudophilia form vacuole. Oh, and here is the food vacuole is formed. Here is the lysosome which take it inside and food enter inside the lysosome and lysosome secrete enzyme and digest it. So that's how lysosome function. Next is vacuoles. Vacuoles are membrane bound space in cytoplasm and it's made up of single membrane which is called tonoblast. Okay, it contains water, sap, excretory product and other materials which are not useful for cell. So in plant cell, central vacuole is found. Okay, so this vacuole store water. Okay, also help to increase cell size. How? See, suppose that this is plant cell, here is the vacuole, this water enter inside the vacuole and then this vacuole push plasma membrane to increase in size. Okay, so the cell size increase. This vacuole take 90% space inside the plant cell. Okay, also it stores some ions like potassium ion, chlorine ion. So these ions are stored against concentration gradient, so active transport take place. Okay, some pigments, waste product also store inside this vacuole. So what is the function of these vacuoles? In protist, food vacuoles are found like amoeba. It maintains osmotic pressure and turgidity. In amoeba, contractile vacuoles for secretion. See, suppose that this is amoeba. Amoeba lives in water. So much water enter inside amoeba due to diffusion from higher concentration to lower concentration. So because of this extra amount of water, the amoeba can be burst. So here contractile vacuoles are found. These vacuoles store the excess water, okay, and secrete it on the surface of the membrane. That's how it maintains the osmoregulation inside the amoeba. So let's see this whole endomembrane system in one picture. See, this is a cell, okay? This is nucleus. This nucleus have endoplasmic reticulum, okay? Endoplasmic reticulum have ribosomes, okay? These are the ribosomes. Ribosome produce protein, okay? Suppose that this is protein. This protein enter inside the ER. Here, initial modification take place, okay? Then these proteins secrete out in the form of vesicles near to the ER. Golgi body present, okay? So this vesicles will attach to the membrane of the Golgi body, then it enter inside the Golgi body. Here the modification, majority of modification plus packaging is done, okay? Packaging is done, and then these proteins secrete out in form of vesicles. This is cis phase, which receive the vesicle. This is trans phase of Golgi body, which secrete the vesicle. Most of the vesicles go outside cell, okay? Some of them are used inside the cell. Some of them special vesicles formed by Golgi body turn into lysosome, and some of them turn into vacuole, okay? So that's how the endomembrane system is work, okay? And always remember that modification is done in both inside ER also, Golgi body also, but majority of modification of protein is done inside the Golgi body. So the topic is done. I hope you like it. Please like, share, subscribe my channel. Thank you.